Welcome to the Francis Crick Lecture, which is given each year for achievements in the fields of genetics, molecular biology, and neurobiology, in which Crick worked. As many of you know, Francis Crick was, amongst many other things, co-discoverer of the structure of DNA. This lectureship was endowed by Sidney Brenner, one of Crick's close associates in Cambridge, and given first in 2003. I'm John Scahill. I'm Vice President and Biological Secretary of the Royal Society, and it's my pleasure tonight to introduce and congratulate the 2017 Francis Crick Lecturer, Simon Myers, Professor of Mathematical Genomics in the Department of Statistics in Oxford University. The award is made for Simon's work in population genetics in man and other species, and specifically for his studies of the patterns of recombination in these different species. Please welcome Professor Simon Myers.
Thank you. So good evening, everybody. I want to start by thanking Sir John for that lovely introduction, and also want to thank the Royal Society for giving me the chance to speak to you tonight and giving me this award. I am completely humbled to receive it. So what I'm going to talk to you about tonight is a lot of very collaborative work that combines work on using statistical inference and also experiments. And I want to talk about three basic biological processes: meiosis, recombination, and speciation, the formation of new species. And especially I want to talk about the connections between those processes, which may not be obvious at first. And I hope by the end to show you that by thinking about the connections between these processes, we learn more about how each one works. So we live on a very richly endowed planet. There's a huge amount of wonderful diversity. Here's a chimpanzee, our closest sister species present on Earth at the moment, and all of this diversity is the result of literally millions of different speciation events that have given us all these rich species. And speciation can actually happen in lots of ways, but fundamentally it always involves one group splitting up into two groups that become reproductively isolated, for example by geography, and then through time these populations start to evolve separately, and genetic differences arise between them. And as that process continues, if they're separated for long enough, what can happen is, even if they come back together again, either they don't breed together, they don't hybridize, or those hybrids have problems. So this is the sort of basic rule, but actually at the molecular level, figuring out what genes are involved in those problems arising, we don't know so much. Really, there's only a handful of genes for speciation that have been identified. And in fact, if we look at mammals, though we have lots of species of mammals, only one specific gene for speciation has ever been found in mammals. And this was by pioneering work led by Jiří Forit's group in the Czech Republic. Over 30 years, actually finally found this gene. I'm going to talk lots about it tonight. But first, I want to sort of talk about the system in which that speciation gene occurs. And it happens in house mice. So, the common everyday pest that comes into your house, often in winter, or your barn. Now, within Europe, there are two different species of house mice, or rather subspecies of house mice. They're called domesticus and musculus. And they've been separated for about 1 million years, so that they're nearly 1% different in terms of their genomic sequences. Now, each of these two subspecies has an exemplar lab strain, shown up here, inbred lab strains, and they're called PWD, which represents the musculus subspecies, and Black 6, which represents the domesticus species. And these guys are actually thought to be in the process of becoming separate species, because when you cross these mice, what you find is the offspring, the male offspring, shown here. If the mother is PWD, they're healthy, nothing wrong with them at all, except they're totally sterile. And if you do the cross the other way around, so that the mother is a Black 6, the males are still only semi-fertile. And when this kind of thing happens, it's thought to be, in some sense, the end game of speciation, because these subspecies have sort of, if this happened in all the mice, they can no longer exchange genes. They're completely split, and they have to evolve independently forever. Right. And this mouse is a great model because, as we'll see, we actually know one of the genes underlying this infertility. But actually, hybrids occur very widely, naturally in nature. So here are some examples of hybrids, and that's a Prius. And many of them, including the Prius, exhibit similar features in that they often have infertility. So these are all healthy. A mule is a cross between a horse and a donkey. I think you could probably guess what the parent species are for the wolfin and the donkey. And so on, this is a savanna cat, often used as a pet. In most of the examples shown up here, these hybrids are sterile, especially the males. So we might wonder, how does that happen? What's going on? So if we actually look at these mice as a model system, their problem is, in terms of infertility, their males simply don't make any sperm at all. And their problems arise during a process called meiosis. And this is where one makes a sperm or an egg cell within the body. And the basic thing, the basic purpose of this is to allow passing on from one of your parents of a single chromosome to you as a child. Right? So I think we're all familiar with the fact that our chromosomes come in pairs. I have 2 copies of my chromosome 1, for example, and I inherit one of those from my father, from a sperm cell, and one of those from my mother, from an egg cell. And those cells are produced during meiosis. So one special thing about meiosis is it's a cell division that reduces the DNA content by half. The second thing that's special about meiosis is that actually my father doesn't just pass on one or the other of his chromosomes to me. What I get is this beautiful, intricately constructed mosaic, and that means that I'm sort of unique as a person. I get a unique combination of DNA. And this mosaic is also constructed in meiosis, and the places where one chromosome switches to another, like here, are sites of what's called recombination. And I'm going to need to talk to you, as this evening goes on, about sort of how recombination works in more detail. And a key thing that has to happen during meiosis for both passing on a single chromosome and recombination to happen is that the chromosomes have to pair up. And so this image is greatly simplified, but basically shows what happens at the beginning of meiosis. So at the beginning, what you can see is that here's a cell, and this is the nucleus, and DNA is in a big tangle all over that cell. All right, it's all over the place. And then something sort of magic happens. As meiosis goes on, what happens is the copies of each chromosome, shown here in blue and red, pair up. So this might be 2 copies of chromosome 1. They align, as if by magic, and that allows them to swap genetic material and recombine. And it also allows, later, because these things are paired, they get pulled apart separately, like this, as is shown here, and then ultimately the cell divides and they end up in separate cells, and that's how we ensure we pass on exactly one of the copies to our offspring. So pairing is really key. So how does pairing happen, and how does recombination happen? Well, the key events for this are things called double-strand breaks. So double-strand breaks happen at the very beginning of meiosis, and actually it's kind of counterintuitive to me. They're what they sound like. At the beginning of meiosis, instead of sort of starting by pairing your chromosomes, what you do is you break them up into lots of tiny little pieces by inducing these breaks. And this is essential for chromosome pairing, but where these breaks happen in the genome later become these recombination events. And so here's a little cartoon of how it happens. There's a gene shown as a pair of scissors here called SPO11 that was discovered by Scott Keeney's group. It's actually present very widely across nature. And this is present in lots of copies within a cell. This represents sort of part of a cell. And these 2 parallel lines here, I'm going to show diagrams a bit like this throughout the talk. Each of those represents a chromosome. So these might be the 2 copies, say, of chromosome 1. I'm not going to bother showing you all the other chromosomes. And so SPO11 just comes along, and 2 different molecules each cut one of the DNA strands, as shown in the circle. And then as part of the processing, SPO11 is removed. And what you end up with is these little holes all over your DNA. So why do you make these little holes? Well, the idea is that mechanistically, these things can now go and search for the corresponding position on the homologous chromosome. This chromosome over here, they need to find this place. And once they do that, they can repair themselves. But they can also then pair up. And one can see sort of under a microscope how this pairing works. So what this diagram shows you is a single cell, the nucleus of a single cell here. And shown in red are the places where double-strand breaks have happened at the beginning of meiosis. They're stained. And what green little segments here show you is pieces of chromosomal structures. And so you can see that they're all over the nucleus. It's very disorganized. But as meiosis proceeds, things sort of start to get more and more organized, so that by this packaging stage, and this is the center of a mouse nucleus, what you can see if you count these is that there are 19 neatly paired chromosomes, these little sausages. So this represents 2 chromosomes attached to each other, with these double-strand breaks still along their length. All right, so in short, we start with double-strand breaks. That helps the chromosomes to pair, and then eventually those positions become places of recombination, this swapping of DNA. And actually only about 10% of breaks repair by what I showed you a moment ago. This is called crossing over. And they do that by kind of forming points called chiasmata, where the chromosomes physically attach to one another. Okay, so we need these breaks to get recombination. So as it says at the top of the slide, recombination actually seems like a crazy thing to do. You deliberately break your genome, then you have to fix it. And in fact, it does have lots of risk. So there's infertility if we don't make breaks at all. And a major consequence of recombination going wrong is that if we don't get at least one of these crossover events per chromosome, that often leads to aneuploidy, passing on the wrong number of chromosomes. And diseases like Down syndrome come from having, in this case, 3 copies of chromosome 21. But also this aneuploidy, which can also happen if recombination events are in the wrong place, is thought to affect up to 30% of human oocytes and is the commonest cause of miscarriage in humans. Now, apart from simply passing on the wrong number of chromosomes, if chromosomes pair up in the wrong way, that can rearrange your genome and leads to diseases like Charcot-Marie-Tooth disease. So it's a risky process. Why do we do it at all? Well, the answer is pretty simply, recombination is widely accepted to be the key benefit of sex. Okay, so we actually have a lot to be thankful for it. And so why is it of benefit? Well, the reason is it shuffles variation. So if you think of these blobs as representing mutations on the chromosomes of one of your parents, recombination means you get a new combination of mutations. This is why you're unique. You're something that's never been seen before. And this generating of genetic variation allows natural selection to happen much more favorably. And so the sort of basic idea is if we didn't have recombinations, our mutations would be in fixed combinations on particular backgrounds. Recombination scrambles the backgrounds that mutations are on. And so we can sort of choose the best combination of mutations to pass on to the next generation by natural selection. And that's good in terms of both helping good things evolve and preventing deleterious or bad mutations from evolving as well. And so one of my other passions, apart from what I'm talking about today, is a study of human ancestry. And in that work, we've developed methods to understand genetic differences among people. And those methods directly infer where recombination has happened between people and use the combinations of mutations that we see to show, in this case, that there are genetic differences between people on fine scales within the British Isles. And in many fields of genetics, recombination is critical. So, for example, we need recombination to narrow down where disease genes are when we're doing disease gene mapping. All right. So for this and lots of other reasons, my group and collaborators have been very interested in studying recombination. And almost the most basic thing we could ask about this process, and this example is for chromosome 12, is where along the genome does it happen? And the answer to that turns out to be rather interesting, in that you might think that recombination might sort of happen reasonably evenly along the chromosome, but actually it doesn't do that at all. Instead, it clusters into lots of hotspots across the genome. And so what the black line shows is work we did ages ago now, where we use variation patterns to estimate the recombination rate along this chromosome. And all I really need you to notice about this, that's rates in black, is that it's very, very spiky indeed. So most of the recombination happens in a tiny fraction of the genome. These hotspots where recombination clusters are shown along the top. And our contribution, I think, in this work wasn't really to discover hotspots. Alec Jeffrey's group and others have done pioneering work to show us there were some hotspots in humans, but by mapping very many of them, we're able to go on and look at the DNA sequence within those hotspots to work out another obvious question, which is there some code for why this place is a hotspot and say this place here isn't? And what we found when we did that, later at least, is that there really is a code for where recombination hotspots happen. So if I sort of walk along the genome, I might observe a stretch of DNA where the first base is a C, the second base is a C, the third base can be anything, and so on. If I see this particular combination of DNA bases, say here along the genome, that place is very often a recombination hotspot. So if you like, this is called a sequence motif, but it represents a code for where these things lie along the genome. And at the point where we discovered this was the first time that anyone had noticed or been able to see that such a code exists in eukaryotes. But since then, lots of work's been done in other species too, and this pattern is rather general. So once we sort of had this code and these hotspots, we understood pretty quickly that probably what was happening was that there was some gene in the genome that reads this code and probably then drives recombination. And eventually, we identified it, and this gene is called PRDM9. I'm going to say plenty about it in the rest of the talk. And this gene turns out to be sort of the key to positioning nearly all of the recombination events that happen in people and also in mice. So you see where it is in the genome in these two species. And actually, over 98% of hotspots are positioned by binding of this gene. And I should say that we and other groups led by Ken Pagan and Bernard Damassi found this gene at the same time. The other two groups were working in mice, we were working in humans, and we found it by taking hundreds of transcription factors and predicting bioinformatically what they would bind. And if I'll step back a second, looking for a match to this sequence motif, and PRDM9 came up. But now this gene is known to operate in many species, and it's present across mammals in general. But interestingly, there's an exception. Dogs and canids don't have this gene. They do have hotspots, but they're differently positioned. I don't know why man's best friend is awkward in that way, but they are. OK. So how does PRDM9 actually recognize this code? Well, the key region of the gene for this talk is going to be this array of so-called zinc fingers. All right, so when PRDM9 is made in a cell, it basically folds into a specific shape where each of these rather similar fingers folds into a shape a bit like a finger, so it looks like this. And then each of them, oops, sorry about that. Each of these zinc fingers contacts 3 bases of DNA, and so when they bind together, they can identify this hotspot code, as we see here. And actually, we've more recently done experiments led by Nick Altamoser, a former student in my group, where we directly measured in human cells where PRDM9 binds. And you see it's a beautiful match to this code we found looking at the hotspots. And PRDM9 has lots of other interesting domains that have been studied by lots of groups around the world, but I won't go into those today. All right, so just to summarize what I've said in the last few minutes, how does this gene really work? Well, during the first part of meiosis, we make lots of copies of PRDM9, and it has this structure here. If we look at a piece of DNA, it contains matches to the binding motif, and then PRDM9 goes along and binds those places specifically in the genome. And when it binds, it puts up little signposts. That's what these balloon-like things show, basically saying, I bound here. And it does that by modifying the histone code, the DNA packaging Again, we won't need to go into that. And then somehow, by a mechanism that we really still don't understand completely, what happens is SPO11 is recruited and the recombination machinery is recruited and breaks the DNA next to where PRDM9 has bound. And because it always breaks DNA near to these motifs that PRDM9 binds, that results in recombination hotspots. So we only get recombination near these positions. All right. So a kind of amazing thing about PRDM9, the reason I've been talking about it in a way, is it turns out also to coincide exactly the same gene is the one that's the speciation gene in this system of mice that I talked about before. So PRDM9 also is the only gene that we know in mammals that causes infertility in hybrids. And it doesn't just do this in this particular system that I've shown you, actually it influences fertility more generally across other hybrids of mice as well. And so we got very interested, and we're still working on to a degree, in the question of, why is it that this happens? What goes wrong in the hybrids? So one thing that we do know is that these PWD and black 6 mice differ in these zinc fingers of PRDM9. And that somehow drives the hybrid sterility. These differences in zinc fingers are what determine whether offspring mice are infertile or fertile. And that might be sort of surprising if you think about it for a second. So recombination is a fundamental and essential function, but yet if PRDM9 zinc fingers are different between two species, it must bind different things, and that is in fact the case. And so it may be even more surprising to learn that PRDM9 isn't just sort of different in these mice, it's actually the fastest evolving gene in the whole mammalian genome. And specifically, the zinc fingers that determine where the hotspots are different in every species that's ever been looked at. So here it is in some of the great apes. And that means that we have different binding motifs. Here's the human Eurasian motif that I showed before. But if we look in mice, PRDM9 recognizes quite different sequences. So recombination hotspots in black 6 happen near this motif, in PWD near this motif. They're different. If we look in an Asian leopard cat, we've inferred a motif that's different again. And then even if we look at species very closely related to us, recombination hotspots in humans and chimps are in totally different places. And even within humans, if we look in people from Africa, 40% of people in Africa have recombination hotspots that are marked by a different motif. And we still don't completely know why this landscape of hotspots, though it's as a feature conserved across many mammals, individually is very fast evolving indeed. But as we'll see, that fast evolution does relate to this hybrid sterility. All right, so how does the hybrid sterility work? To actually figure that out, we need to sort of burrow a bit deeper into what goes wrong in these infertile hybrid mice. So remember, they're totally sterile. They fail to produce sperm. But actually, what really goes wrong is rather early in meiosis, when chromosomes should pair. What they actually do is they fail to pair chromosomes. And so just as before, each of these images shows a single cell. And this is a sort of parent mouse, the black 6 mouse, and where the only difference from before is now that the red, sorry for those who are colorblind, it's difficult to use different colors. So I sort of used the pointer to help, hopefully. In red, chromosomes are stained if they fail to pair. So in a normal healthy mouse, at this stage of meiosis, the only chromosome that is in red and isn't pairing, that's the XY. And they're different, so they can't pair. All the other 19 are neatly paired. But if we look in these hybrid mice that are sterile, here's what we see. We see lots of chromosomes in red, and they're helpfully marked by the arrows, even if you are colorblind. And in a way, sort of sadly and touchingly, some of them, because they haven't found their partner to pair with, have sort of tried to pair with themselves. And so they form these circles. And so the question becomes, what is it about PRDM9 binding that's affecting this pairing? And we may hope that this is a sort of window into how it should work. Okay, so how it should work, we actually, as we'll see, we don't really know. So can we sort of learn something about that question? And actually, chromosome pairing is a miraculous thing. So it's sort of hard to conceptualize even how it works. But I've shown you sort of that there are these double-strand breaks, and recombination involves sort of identifying exactly the corresponding position on the other chromosomal copy, the homolog. Well, there's only one of those positions in the whole genome, so what you've got to do is sort of find your corresponding place among the 5.6 billion possibilities all around the cell. And so if you actually blow up or expand the DNA within a single cell so that it's stretched from London to New York, so your genome becomes very, very long, that's equivalent to finding something the size of a single grain of sand along that distance. So it's somewhat miraculous that it ever works at all. And exactly how you managed to find that grain of sand between London and New York has been basically unknown. But what we do know is that it's extremely robust. So different mammals have very different numbers of chromosomes. And so here are two extremes. And the winner, in terms of mammals, in terms of how many chromosomes there are, is this Plains rat from Argentina. It has 51 pairs of chromosomes. And they manage to find each other and pair and get put into offspring correctly basically every single time. Okay, so how does that work? And to investigate this, we studied a mouse that Ben Davis, the Wellcome Center Human Genomic Transgenics Core head, had produced. And this mouse is identical to the black 6 mouse that's involved in this hybrid infertility, except that the PRDM9 zinc finger array that's normally found in mouse was replaced with that in humans. And the initial idea was to try and study recombination in humans using the mouse. So what happens when we do this? Well, the resulting mice are actually completely normal and healthy, and they're completely fertile. But they have, as we actually expected, completely reprogrammed hotspots. So there are these sort of 16,000 new hotspots in this humanized mouse don't overlap at all with the ones in the original black 6, more than you'd expect by chance. And then we had this sort of bright idea, at least eventually, of looking to see how this behaved in this hybrid system. And so compared to the original sterile infertile hybrids, we bred these mice where we placed this black 6 with the one carrying human PRDM9. And the question is, what happens? And so just remember that sort of in the infertile hybrids, there's all this failure to pair chromosomes. Now, if speciation is the result of the accumulation of differences, what we've done by using a human mice is sort of certainly not to reduce the number of differences, so we'd expect, or we did expect, that these hybrids would also be sterile. And so we were a bit thrown by this fact. When we looked at them, when we have these humanized mice, they're completely fertile. They're just as fertile, these guys, as one of their parents. Okay, so we completely and unexpectedly fixed this sterility, so we call this a humanized rescue hybrid. And actually, in some ways, it gets even more extreme. So if we take this humanized mouse and we now cross it with a completely different species of mouse called Spretus, so Spretus mice live in Spain and North Africa. So normally, crosses between this different species of mouse and black 6 are completely sterile if they're male. But once again, we restore sperm production, although admittedly, the sperm are somewhat abnormal, but we're able to get offspring from them. And so what this means is that in some sense, we're able to reverse these speciation events just by putting in human PRDM9. And actually, it's not only the human allele that does this. If we put in different zinc finger arrays, largely the same thing happens. And I think one way to conceptualize what's happening here is that by putting in the human allele, or any other allele, we're putting in something that hasn't been tested by natural selection in mice. So it's just the same as any other random newly arising allele. And so what this really says is sort of any random newly arising allele that appears in mice could restore fertility to the hybrids. And the thing that's surprising about this is it tells us that although we think of it maybe as an endpoint of speciation, this hybrid infertility can be reversed. Okay, and it could reverse naturally as well. On the other hand, because at the moment hybrids come with a large cost, it might be that other mutations could occur in the meantime to prevent forming the hybrids, which genetically are sort of wasteful for the species, and that might lock in speciation. So there's sort of a race going on. Okay, so now we have this sort of a setting where we either have infertility or we don't in hybrids, and we might wonder what's going on. And we know in some sense that what's going on is controlled by where PRDM9 binds along the genome. That's the only difference between these two mice, and so we measured that. Okay, so for those of you who are not experts in all these techniques, don't worry too much. What we did is we used what are called chip-seq experiments to measure along the genome binding by PRDM9, and also double-strand breaks, the breaking of the genome where PRDM9 binds, using clever techniques first developed by Dan Camerini-Otera's lab. And this work was done as a collaboration jointly led by my lab and that of Peter Donnelly. And all you really need to know about chip-seq is what it does is it uses antibodies to sort of pull down stretches of DNA near places that are bound by some protein, in this case PRDM9. And then you can throw those pieces of DNA into a sequencing machine, you sequence them, and then when you map them back along the genome, you find out where the protein bound. And you get a readout like this, where the peaks shown by the arrows correspond to hotspots containing these motifs. And so we did that in both these hybrids. Now, actually, we need to look at the results in a little more detail than just that single readout. And that's because in these hybrid mice, firstly, there are two chromosomes, two of each chromosome, one BLAC6 and one PWD, because those are the parents. But also, there are two different PRDM9 types, the PWD and BLAC6 PRDM9 types shown up here. And either of them could bind either chromosome in some combination. And actually, we can figure out which is which. So firstly, because these genomes differ at mutations, from the raw sequencing data, we can find out which chromosome PRDM9 tends to bind and break. And so looking across these hotspots, we get numbers like this. And so some hotspots are bound and broken equally on this chromosome and this one. We call those symmetric. Whereas for others, if all the sequencing reads come from this chromosome, we know that's the one that PRDM9 binds and breaks. The second thing that we can do is to then look at the DNA sequence of each hotspot for PRDM9 motifs. And depending whether we see the binding motif of BLAC6 or PWD, we can tell which PRDM9 type controls that hotspot. And so ultimately, we get to identify which allele is happening. So this hotspot is controlled by BLAC6 and is symmetric, whereas this one here is controlled by PWD and is asymmetric. It's only active on the BLAC6 chromosome. All right, so we did this across the whole genome. And for those of you, I'm a statistician, who like to look at data and graphs, here's just about the only graph I'm going to show. It shows the results. And what's on the x-axis is how much of a signal comes from cutting one particular chromosome, this BLAC6 one. And so symmetric hotspots should be 50-50. But you see there are very few. In fact, nearly all the hotspots cut only one chromosome or another. And that was initially a bit puzzling. And there's also a pattern that we can see. Those hotspots controlled by PWD, they're in pink, tend to be on this side. And that means that the BLAC6 chromosome is cut. Those controlled by BLAC6 are on this side, so the PWD chromosome is cut. All right, so if that's a lot to take in, the sort of diagram summarizes what happens in this mouse. So essentially, each of the PRDM9 alleles, say the PWD one, binds only the other genome, or mainly the other genome. And so shown in circles here are places where, blue circles are where BLAC6 PRDM9 binds. It mainly binds along the PWD chromosome, and the corresponding positions on the BLAC6 chromosome are not bound. The converse happens for the other allele, and there are very few places like this where PRDM9 binds both the homologous chromosomes at the same place. So what happens if we humanize it is that we lose all of these BLAC6-controlled hotspots because we replace that PRDM9 with the human one. So we can look at the new hotspots that we get, and they look completely different. Now, most of those are symmetric, And so again, referring to the diagram in this mouse, how PDM9 works looks like this, and it's very, very different. So PWD-controlled hotspots are just as before, but now BLAC6 PDM9-controlled hotspots like these tend to be active on both the chromosomes at the same places. And this led us, it seems pretty obvious in hindsight, but it took us quite a long time to figure this out. But eventually we got the right answer, which is it led us to think maybe that these sites where PDM9 binds both chromosomes at the same place are used to pair the chromosomes. And that would explain what we see. So in animals like the humanized one, where PDM9 binds both chromosomes at the same place, most of the time chromosomes pair fine. In the parents of these mice, like the BLAC6, there is no genetic variation. So PDM9 always binds the same chromosomes at each place, and pairing is just fine. But in the infertile hybrid only, PDM9 is binding only one or other of the chromosomes, not both, and pairing fails. So it explains what we see as a model. As I'll show you in a second, we have lots of evidence that this is the right idea. So if this is the case, actually, it means that PDM9 does something we didn't before suspect. So what we knew is that it binds one chromosome and helps that chromosome to break. But what this would mean is that what actually happens on the other chromosome, the homologue, is just as important in determining how things work. So how does this asymmetry pattern, which seems kind of bizarre, actually arise? The sort of high-level message is it arises because hotspots are self-destructive. In some sense, they're suicidal. And this happens as an evolutionary process. Okay, so if we actually look at a single asymmetric hotspot, say it's controlled by BLAC6 PDM9, then as I've shown you, most of the time, that hotspot has an active motif that PDM9 can bind on the PWD chromosome. On the BLAC6 chromosome, PDM9 doesn't bind because of mutations in that motif. So more than 90% of the time, what we observe is that the ancestor had binding by PDM9, and a mutation has evolved along the BLAC6 lineage that stops binding from happening. And for the PWD, we see the converse. And so what this means is that hotspots are being lost very rapidly through time, more rapidly than we'd expect for random mutation, along the particular lineages where they're active. So in the ancestors of the BLAC6 animals, the BLAC6 PDM9 motif has been going on, and that's caused this rapid change, and conversely on this lineage. And that's why we see the asymmetry. So why do we get this rapid mutational process? Why does PDM9 erode hotspots? Well, it's actually kind of technical, but here's a simplified version of what happens. So if you imagine that originally you have a PDM9 binding site in a certain population, shown by the red rectangle here, then a random mutation can happen in some animal that stops PDM9 from binding. So this just represents some mutation. And now what then happens is that when double-strand breaks happen at this hotspot, PDM9 will only be binding this chromosome, so this chromosome will be the one that breaks, and not this one. And so how is this double-strand break repaired? Well, it's repaired by recombination, and that results in using this homologue to fill in the gap. And when that gap is filled in, that means it passes on this particular mutation. So the end process of all of this is that mutations that disrupt binding by PDM9 tend to be passed on to offspring much more than one would expect by chance. And over time, that leads to the rapid accumulation of these mutations and this asymmetry. And it's probably important to note that this doesn't just happen in mice. We see this very widely in species with PDM9 because it's a mechanistic consequence of recombination. And for example, it also happens in humans and on our lineage as well. All right. We're actually very confident that this idea that symmetry is important for binding is actually correct. So what we did to test this idea is that we gathered data like the data I've shown you, ChIP-seq data about PDM9 binding across multiple mice, including the infertile and this humanized rescue in purple that I've just shown. And from those data, we quantified for each mouse whether PDM9 binds symmetrically or asymmetrically. So we can order them, as you see down here. And then we tested how that related to the fertility of each mouse. And so, for example, we use various fertility measures, but one of them is how often the chromosomes pair properly, which I've shown you on several slides. And what we see is a basically perfect correlation. So the more symmetric PDM9's binding is, the more fertile is the mouse. And we actually see exactly the same thing across 36 extra mice, not shown here, which differ either in terms of what PDM9 they copy, that can affect this symmetry measure, or in terms of they have the same PDM9 alleles, but they have different genomes, and that can affect the symmetry measure too. Whichever one we use, we always see essentially this complete correlation between fertility and how PDM9 binds. And so there's really overwhelming evidence that this does control things. And it also tells us that fertility isn't really an on or off thing, it's a spectrum. So these mice are varying in how well they do meiotically. And so this total infertility is just one end of a spectrum. So hard speciation is one thing that can happen, but you can have more subtly reduced fertility. All right. So the last thing we've tried to do, and work this is still going on, is to sort of figure out why this happens. And one thing that we can do is to sort of zoom in to the level of an individual chromosome and ask, does that chromosome pair? So if we look at chromosome 16 in a typical infertile hybrid, actually that mouse fails to pair this chromosome about 22% of the time. And actually the sterility is caused from the cumulative effect of failing to pair individual chromosomes adding up over chromosomes. And what we decided to look at in collaboration with Yi Forate, who's a person who found the role of PDM9 in speciation, is what happens if we modify this chromosome by introducing a small number of hotspots where PDM9 binds to both homologs symmetrically. So we put in just a few symmetric hotspots into this chromosome, and when we do that, it completely normally pairs. And so what this means is that the whole chromosome can pair up if there are just a small number of these symmetric hotspots on it. And what that suggests is that really this pairing system is very efficient as long as we have some symmetric binding. And we can actually go down to the level of individual double-strand breaks. And so we've got actually evidence from lots of sources that if we look at an individual double-strand break, shown here, it can either happen at a place where PDM9 binds the homolog or where it doesn't. And in fact, that break takes much longer to repair if the homolog isn't bound by PDM9 than if it is. And an interpretation of that is that basically how this works is that every double-strand break is a chance for pairing chromosomes. It needs to find its homolog to repair. And that process works much better if the homolog is bound, because what happens is instead of sort of searching the whole genome to find your match, you just search among other PDM9 binding sites. So you think, my break happened at a recombination hotspot. The place I need to repair is a place like me, so that should be a recombination hotspot as well. And so I just check those places along the genome that have these signposts that say PDM9 bound. And I'll just finish by sort of talking a little bit about what this might mean. So why would we do this? Well, the reason is, instead of checking the whole genome, if we only check PDM9 binding sites to pair chromosomes, we can get several thousand-fold efficiency because only a tiny amount of the genome lies in these hotspots. And so maybe that's why you can sort of find that grain of sand between London and New York. You don't check the whole of the way from London to New York. You just check the relatively small number of places where PDM9 binds. It's like losing something and only looking in the likely places you might have lost it. Okay, and that raises the idea that actually maybe this is the purpose of hotspots. So a nice question is, why does recombination happen in this interesting and complicated way at all? Why doesn't it happen evenly along the genome? Well, one reason might be if kind of most or all species with hotspots, the purpose of those is to refine this search and make it easier to find your homolog. And it's notable that actually this could sort of not just depend on having PDM9. Lots of species without PDM9 still have hotspots. And generally in those species, pairing still requires double-strand breaks to happen. So hotspots might be a general means of pairing up chromosomes. And maybe the exceptions prove the rule, because in a small number of species like these Drosophila flies and these C. elegans worms, double-strand breaks actually happen after pairing, so they're definitely not needed for pairing. So far, when species like that have been found, you don't need recombination hotspots. In fact, recombination hotspots don't occur. So maybe this adds evidence to the idea that that's what hotspots are for. And so what about speciation and infertility? Well, that seems to arise from the fact that this process of pairing has an Achilles heel. And the problem is that we have these beautiful signposts that mark where recombination hotspots and help pairing, but those signposts evolve really quickly and disappear from the genome. And so when one gets hybrids, or when two populations get separated, those signposts start to change, and then PRDM9 can change and all the signposts become different. And then when they get back together, the chromosomes can no longer find each other. And if this happens more generally, and there's no reason to think it doesn't happen more generally in mammals because we know that erosion is a rather widespread thing, it means that this kind of speciation, this hybrid infertility arises not because of some sort of positive evolutionary event, but just by accident. Because the species was separated for long enough time, this hybrid infertility can arise. And sterility may be just one extreme. We might see actually a lowering of fertility is a much more common outcome. And finally, this mechanism is kind of different to classical models of how a gene works in speciation, like this model here of Dobzhansky-Muller incompatibilities, where one often thinks of several genes evolving differently between two populations, so that when they come back together, they interact with each other in negative ways and basically mess each other up and stop them doing their jobs. This system doesn't require multiple genes. There's only one gene here, PRDM9, but it changes, it evolves, and then interacts with the whole rest of the genome to cause this hybrid sterility. So whether other speciation examples are like that, whether there are other mammalian cases where PRDM9 plays a role, remains to be seen. So that's all I want to say, apart from to thank all of the people that have been involved in the work I've talked about today. So firstly, I'd like to thank my group who make me smile every day. And these members of my group have been directly involved in the work I've put up. And I want to thank key collaborators over the years of my career. So Peter Donnelly co-led lots of work I talk about today, but other important collaborators are Yeji and Gil. Lots of the beautiful images I've shown today were produced by Daniela and Kath, who are sat down here at the front. And the mouse work was led by Ben Davis. And I also just want to thank the people who sort of put me up here today, starting from my undergraduate lecturers, Bob, my DPhil supervisor, and Peter and David, my postdoc supervisors. I just want to thank them from the bottom of my heart, really, for sort of setting me on the course to doing this work that I enjoy so much. And finally, thanks to all of you for listening this evening. Thank you.